So we can now move on to our third talk for this session. This will be held by Marianna Mitsioni. She's a PhD student from the lab of Alexandra Radenovic at the EPFL in Lausanne. They are also mainly working on biological nanopores. She's mainly using Fred techniques, but also electrophysiology. So Jan also gave an excellent introduction about the fundamentals, and we will now see another example of coupling optical and electrical readout.
Hello everyone, thank you so much for the introduction. I am Marianna, and today I will talk about fluorescence-based approaches to study biological nanopore mechanisms. So, no surprise, I work with pore-forming toxins. So these proteins that in some cases they are expressed as monomers, and in the presence of a membrane, they oligomerize into a pre-pore complex, and then in the end they form an ion channel in the membrane. My personal favorites are the beta pore-forming toxins and especially aerolysins. And as we heard before, these pores have been taken out of their biological context the last years, and they are used for sensing. So in a typical experimental setup, we have two electrolyte filled chambers separated from a lipid bilayer where we insert a single pore and we're driving voltage to see the current. If no analyte is present, we get a value of open pore current. But when we introduce the analyte and the analyte is translocating through the lumen, we're getting these drops in the current, which we analyze to get valuable information. So us as well, we are trying to combine this with single molecule optical readout and do fluorescence imaging at the same time. Basically add the eyes to the system to address some key limitations. That's why today I will focus on two things. First of all, how we use fluorescence in the lab to study a mechanism of beta-barrel pore-forming toxins, but also some methods we have been developing in the lab to expand what we can see with these pores. So for the first project, we aim to investigate dynamic properties of these proteins, and especially to fundamental nonlinear behavior, open pore rectification, and gating. To understand these behaviors a bit better, we go back to our basic setup where no analyte is present, and we apply a constant voltage. The first thing we see is that the fact that the absolute values of current at plus 100 millivolts and minus 100 millivolts are different. This is called rectification. We can also see it in the IV curves, because if it was not present, we would have an ohmic behavior. And the second thing we see is that at one polarity, at negative polarity, at a random time point, we see a drop in the conductance. This is stochastic and it's called gating. And even though it is reversible, it is a problem for sensing. So we thought that if we manage to quantify and model these two behaviors, we will be able to engineer better pores for the sensing applications. So let's start with the open pore rectification. We tried many different experimental conditions to see what it depends on, but the first results we got when we focused on the charged amino acids inside the pore lumen. So aerolysin has 13 rings of charged amino acids in the lumen, positively or negatively charged, which form energy barriers, and they actually shape how ions are transported into the pore. And we created a library of 22 mutants where we started deleting charged amino acids from the lumen or combination of charged amino acids. And what we observed was a huge range of behaviors from convex to concave shapes when we plotted the IV curves. And when we deleted all the 13 charged amino acids from the lumen, we also got a pore that had an almost ohmic behavior. So based on the IV curves, we managed to model the rectification behavior based on the position and the kind of the charge we were deleting out. Now for the gating, quantification was a bit more complicated because as I said, it is stochastic. We figured out that it's easier to quantify it if we apply alternating voltage and we plot the IV curves. So you can see at positive voltages, the pores are behaving normally for aerolysin wild type, but at the negative voltages, the pore is fluctuating between the open and the closed state, the gated state. Now, if we average out multiple of these IV curves, we get a smooth hysteresis loop, which is exactly the same thing that we get if we do this cycle once, but with many more pores in the membrane. And through these loops, we can extract the possibility of the pores being open or closed at specific voltages, but also the closing rates. And other thing we observed is that for the wild-type protein that is mostly positively charged at the bottom of the barrel, we observed gating at negative voltages, while when we created mutants, where the pore was mostly negatively charged at the bottom, the effect was reversed and gating happened at the positive voltages. We also created mutants where the pores were gating in both polarities, and also mutants where the pores were not gating at all, at least in the frequency we were using. And going back to our library of mutants, we managed to connect the rectification factor to closing rates and fully model and rationalize gating behavior as well. Then we thought of expanding to other pore-forming toxin families, and we created libraries of alpha hemolysin and MSPA mutants as well. And we saw that the behavior of gating is universal among the three families. But we didn't want to stay there. We wanted to take this also a step further and propose a mechanism behind gating. And what we're proposing is the deformation of the barrel. We think that the electric field puts some forces on the charged amino acids in the lumen, and after some time we get a disordered barrel and we have a reduction of the diameter. The evidence we have to support that is that gating depends on the thickness of the membrane, so thicker membranes suppress gating, but also when I cross-link the pores, making them stiffer, gating was also gone. So from now on, what we are trying to do in order to get direct proof that gating can be attributed to conformational change is doing single molecule FRET measurements, basically detecting conformational changes in the beta barrel by measuring distance-dependent energy transfer between a pair of fluorophores. So our plan is to label pores with a single donor and a single acceptor, then taking the pores to the setup that was described before, when we can do electrical and optical measurements at the same time. And our dream is to build up statistics for FRET efficiency when the pores are gating and when the pores are not gating. So the first problem we met for this project was, of course, the labeling of the pores. We wanted to have heteroligomers where some monomers were labeled and some were not. I found out that the most effective way to do this is to label non-activating monomers with donors, some with acceptor, then mixing with wild type at controlled rations. And I also tried different constructs when the labels were at different positions. We figured out that this mutant was the best to go for, and at least in supported lipid bilayers, we were getting populations that were labeled with different fluorophores at different positions, but we also managed to get some good FRET traces, and by good I mean that the intensity of the donor and the acceptor have uncorrelated behavior, and we also have a single photobleaching steps for donor and acceptor, meaning that indeed the pores are labeled with only one donor and one acceptor. When we tried to take this to the optical chips, of course, we met another problem. We had a lot of electrical signal, but we had disproportionately low optical signal. And we believe that this is because the wild type monomers have lower affinity to labeled ones compared to themselves. So we're getting so many pores that were not labeled. To solve this problem, we added an extra histag to the monomer, but only to the monomers that were labeled. So after activation and pore formation, only the pores that have at least one labeled monomer have a tag. And from this, we can pull with a second purification step only the labeled pores. So with this sample, we actually managed to detect approximately 80% of the pores we would expect based on the electrical signal. And we also have the fret traces. And to be honest, even though we're getting fret signal, we have a lot of problems. One of the main problems we have is that we're not getting the fret efficiency I was expecting by using these specific fluorophores. We're getting much less. And another problem we have is that we don't see single photobleaching steps yet, which makes me think that probably what we are detecting is aggregates and not single molecules. So to answer to your question, we are not there yet in Lausanne either. So this is how far I reached with this project. So if you have any feedback or ideas of how to make this better, I'm super open. And very quickly to the second part, I want to talk about some methods we developed in the lab the last 1.5 years to see what more we can do with these pores. So we wanted to expand our tools beyond FRED, so we thought about FLIM. In a fluorescent lifetime imaging, we're not directly detecting the intensity of the fluorophores, but we are directly detecting the decay. And like this, we can get information about conformational changes, local environment, and molecular interactions. But we also wanted to do single molecule FLIM for thousands of pores at the same time. So with my colleague, Nathan Ronserai, we worked on developing a method for wide field single molecule FLIM, using as the main tool a time-gated SPAT camera that is developed at EPFL. In order to do this, we built this setup. So in the lab, we have a pulsed laser, a green beam that undergoes total internal reflection on a glass cover slip in order to excite only the molecules that are very close to the glass cover slip. And the signal is detected by the time-gated camera that is synchronized with the laser. In order to determine the lifetime of each molecule, first we use the rapid lifetime determination scheme. This requires that the camera has two gates. The first gate, for the first time, the camera is acquiring right after the laser excitation pulse, and that's why the molecules are brighter. While for the second time, we have a delay and the camera is acquiring after some time. That's why the molecules are dimmer. And by calculating the number of photons, the background, and the delay, we can determine the lifetime. So as an application for this method, we've thought of trying to resolve pores that were labeled with different dyes. So we're working with fully formed pores that were on average labeled with a single dye. And from the lifetimes, you can see the distributions, you can see that we can really tell with which fluorophore is labeled each pore. And we can do this for 1,000 pores at the same time with an acquisition time for less than 1 minute. And also, this method offers multiplexing because I'm using a single color excitation, but I can work with 3 dyes. Another problem we have when we image these pores is that things are happening fast. It was shown a few years ago that for alpha hemolysin, it's basically not possible now to capture the oligomerization process. That's why we thought of trying to see how fast we can go for tracking. That's why we used our camera in the lab, but the settings of getting a binary imaging, so images, so single photon images, And we sent data of aerolysin diffusing on supported lipid bilayers to a group in Arizona, the group of Steve Prese, and they actually managed to develop a Bayesian approach to do single photon, single particle tracking. Now our dream is to combine these methods with the mecha chips in order to also maybe capture dynamically the oligomerization of some pores. We have a lot of things in mind that we would like to quantify and ideas of how to do it, but still we are in very early stages. And to wrap up, I hope that I can contribute to the sensing applications with and address some key limitations like gating with modeling, but also to combine our methods to complement some structural insights we have for the oligomerization processes of these pores. At this point, I would like to thank all the collaborators, my lab mates, Alexandra Radenovic and Matteo Talperaro, for supervising me. Thank you for the invitation, and thank you all for your time and your patience.
Thank you so much, Marianna. I was just wondering, I mean, we had this topic before when Jan was giving his presentation. Do you see any influence of all these? I mean, you have all these mutants, and then you have to fluorescently label them. And these labels by nature are quite large. Do you see any influence on the pore as such, on the, like, do you get different multimeric states, for example? Does anything hinder?
Yes, so we first try everything in the not optical chips, and we see some differences in the open pore currents. But we hope that this would not, because we're trying to make the pores gate and catch them in a deep gated stage. So for us, we want to see an ensemble behavior and not really correlate each step to each pore. So for us, we just care about the changes we see both in fret efficiency and in the electrical signal. I don't know if I replied to that.
No, that's fine. Jan?
I mean, of course, I have questions. So first of all, you're saying you have a suspicion that you're looking at aggregates.
Yes.
You would see that also from the diffusion coefficient, no? Is that I saw you had some tracks there. Have you calculated what that would be?
Of the aggregates, or because we have many different populations.
Many different. So it's very heterogeneous.
Yes, it is heterogeneous because I can also not predict where the fluorophores will fall. It can be in monomers that are very close together or very far apart. We are experimenting with linking the monomers with flexible linkers, but to express trimers and tetramers is already...
And in the presence of these things that you suspect to be aggregates, how many pores do you have electrically, typically? I mean, do you have 1 or 2, or do you have 15 or 100?
So, for example, in the video I showed, I cannot see the whole chip because I'm also splitting my field of view. But as you said, you also don't need to see the whole chip because there's a part that is very bad. So we get a signal that would correspond to 75 pores, and in half the field of
the electrical
yes, the
signal.
electrical.
Okay.
And in the optical, in half the field of view, we can detect 31 pores, but not all of them give us good fret signal, right?
But
because
you have 31 like fluorescent dots.
Yes, fluorescent dots.
You suspect them to be aggregates rather than single pores.
This is the confusing part, that I don't know if I actually see aggregates or if there is a problem with the fret signal or actually the protein.
Have you ever tried just reconstituting pores that all have the same fluorophore? I mean, from homomeric labeled ones. And can you look at sequential patching? Because that would be my first idea, that just to do this and then see whether I can see 7 steps, right? Because that would be what you would expect.
Exactly. So this we've done. We have mutants when we actually label at the outer part of the mushroom to not mess up with the electrical signal as well. This was the first thing we tried. And we've tried it. We see multiple steps, but of course not all of them have perfect 7 steps. And again, there we get different populations of intensities.